So we were just on this slide when the bell rang yesterday, and so I just wanted to make sure that we finished it off and that everybody had the things that were on this. I believe I had written down what dominant and recessive meant. No? Okay, then that's where we'll start. So we were talking about Mendel, and basically what we had said is that the F1 plants only showed one trait of the two that were crossed. An example of this would be if we had a plant with round seeds and we crossed it with a plant with wrinkled seeds. All of the F1 generation had round seeds, even though some of the parents had wrinkled seeds. So we get an operational definition of what dominant and recessive means. The dominant trait is the trait expressed in the F1 generation. So if he took two pure plants and crossed them, and only one of the traits showed up in the next generation, that trait was the dominant one. So in my example, that would be round seeds. An operational definition of recessive is the trait not expressed in the F1 generation. And in my example, that would be the wrinkled seeds. And what Mendel saw with the characteristics that he was studying, at least with these pea plants, was complete dominance. This means an individual with both dominant and recessive forms. So that means an individual that has a copy of the dominant gene and a copy of the recessive gene has the same physical characteristics as an individual with two dominant forms. So complete dominance is when an individual with both dominant and recessive forms has the same physical characteristics as an individual with two copies of the dominant form. So just as a little example to show you how this would have looked, there's a little visual representation of this particular cross. Here is my parental generation. Where I took a pure round seed plant. So I put a capital R and a capital R, two copies of that dominant gene. And I crossed it with a pure wrinkled seed plant. I put two lowercase r's to represent two copies of the recessive form. In the F1 generation, the possibilities for combination of gametes are all the same. So all of the F1 generation would have one copy of the dominant form and one copy of the recessive form. All of these have the same physical appearance as the original round seed. Even though they don't have the same gene combination, we're saying that that gene was completely dominant. Even though F1 has one copy of the dominant and one copy of the recessive, they look the same as an individual that had two copies of the dominant form. So that's the F1 generation. He took two pure parents, crossed them, and figured out that some characteristics were recessive and some characteristics were dominant. Does anybody have any questions they want to ask about that part? So now we'll see what happened when he did the next generation of plants after this. So this brings us to a discussion of Mendel's first law, which is the law of segregation. And so just to give us some lead up into this, in the F1 generation, we saw one expressed trait. So even though the parents had two different traits, we only saw one of them in the F1 generation. In the F2 generation, we saw that some had the recessive trait. Now I say some, not a lot of them, not many of them, not most of them, just some of them. In general, in the F2 generation, the ratio is 3:1, and this is the ratio that you will see very frequently. That means 3 out of 4 and 1 out of 4, because there would be 4 total. But that means that for every 3 that showed the dominant trait, there would be 1 that showed the recessive trait. Now, just so we're clear, the way that we got the F2 generation was by taking 2 of the F1 generations and crossing them together. So anytime we're going to the next generation, it means we took 2 from the previous generation and crossed them. So what is Mendel's first law? Mendel's first law talks about genes or alleles being distributed completely randomly during the production of gametes. So his law says that each individual has 2 copies of a gene. Now, I'm saying the word gene, but Mendel didn't actually use the word gene. He used the word factors. So if you're reading something about Mendel, they might use the terminology that he used instead of that. I prefer to use the actual word that we're going to be using in class. So he said each individual has 2 copies of a gene, which he called a factor. These copies segregate randomly during meiosis. So for every characteristic that these pea plants had, the genes separated randomly during meiosis. And each gamete gets 1 copy, so 1 form. Now he uses the word form, and the word form is going to represent something that we'll refer to as an allele. Allele is the word that we use to represent a different version of the same gene. So for example, in pea plants, there was a gene to determine whether the seed is round or wrinkly. The round version is one allele. The wrinkly version is another allele. So the word allele literally means different forms of the same gene. He used the word form, we'll use the word allele, but they are referring to the same thing. Now the last thing, anytime we're representing a cross, the capital letter represents the dominant allele, and the lowercase letter will represent the recessive allele. And that will be consistent throughout all of the crosses that we will take a look at. Now, I just have a couple of comments to make about the ratio. I showed this as a fraction, and fractions are hard to write in a numeric response question. They might be part of a multiple choice. I wanted to make sure that you recognize that 3 over 4 is like 0.75, which is the same as 75%. 0.75 is an easy number to put as the answer to a numeric response because you need 4 spaces, and that conveniently takes up 4 spaces. You need to be comfortable with this, that 1 out of 4 is like 0.25 or 25%. Now, I realize that this is some math, but it's expected that you are able to use fractions and percentages. If you feel uncomfortable with this, please let me know and I will help you practice this. I'll remind you that you are welcome to use a calculator if you would like to. At one point, the fractions will get a little bit trickier. They won't be out of 4, they'll be out of like 16. So you might want to use a calculator just to make sure that you are converting the fraction correctly to a decimal number. Over here, we have just a little representation now of this entire cross. So this and this were on the previous slide. They're just reminding you what the parental and the F1 generation look like. For the F2 generation, we're saying we took 2 of these and we crossed them together. And when we took 2 of those and crossed them together, we ended up getting some organisms that showed the dominant trait and some organisms that showed the recessive trait. These 3 are all the ones that showed the dominant trait because they had at least 1 copy of the capital R. This little guy over here is the one that showed the recessive trait. So this is what Mendel found, and this is his first law, segregation. Basically, he's saying, even though in this F1 generation, you could theoretically only inherit the dominant gene or only inherit the recessive gene, things should segregate randomly. And if we take a big enough sample, there should be some of each possible combination of gametes. Does anyone want to ask any questions here? So I have a little bit more vocabulary that we have to go over, and then I will show you how to do a cross yourself. There are 4 words here that you need to be able to use when you represent a cross. The first word is genotype. So probably you know what the word type means, like what kind, what category. Genotype is the combination of alleles. So if I were to ask you, what is this individual's genotype, I don't want to know what they look like. I want to know which 2 copies of the gene do they have, which 2 alleles do they have. So as an example here, for this organism, AA is the genotype. So I didn't say what it looked like, I said what gene combination it had. Phenotype is going to be its outward appearance. Hello. Sure. Braden, can you see Mrs. Epp, please? So phenotype is going to be the physical expression. So if I were to look at this same example and choose this same organism, I would say that its phenotype is yellow. So I'm not saying anything about what genes it has, whether it has dominant or recessive genes. I'm saying what it looks like. The next 2 words describe what type of genotype you have. So if I had to say, these 2 words are like subcategories of the word genotype. A homozygote, that would be an individual. We could also use this as an adjective and say homozygous. So you'll see both forms of the word. But an individual who is homozygous, or a homozygote, would be an individual that has two copies of the same allele. So if I were to use my same picture as an example, this organism is a homozygote. So is this one. It doesn't say which allele you have to have a copy of. Just says that you have two copies of the same one. So if I take a look at my two choices, one of them is two capital A's, one of them is two lowercase a's. So we would specify and say, is this a dominant homozygote or a recessive homozygote, so that we can be clear which allele is it that they have two copies of. A heterozygote has two different alleles. So on my little example, that individual would be a heterozygote. We don't have to say dominant or recessive, because kind of by definition, you have one of each. So you don't have to specify. But for a homozygote, you have to specify. Otherwise, you don't know if it means two capital or two lowercase letters. So those four words you will see very frequently when we're talking about crosses. Does anyone want to ask any questions about those vocabulary words? So we are finally getting to how to actually do a cross, and some of you might have seen this before. But we're going to talk about making a Punnett Square. Have any of you done this before? A few of you have. So a Punnett Square is our method of analyzing a cross. So when we take two organisms and cross them, a Punnett Square is a good visual representation. In a Punnett Square then, which I've represented down here, there are a few things that are sort of standard. Generally, you will put the male gametes on one side. Very frequently, you will see the symbol for male instead of the word male. And that'll start happening more frequently, especially as we get to the end of this section. And then you would put the female gametes, which you will often see represented with the female symbol, along the other side. It's pretty standard in your textbook that the male goes on the left and the female goes on the top. It's not a rule, but that's typically how things in your textbook would be set up. Now, when you make a Punnett square, you are representing the gametes that are possible for each organism. Basically, you are showing which alleles does the organism have. They might have two of the same allele, they might have two different alleles, but that's what we're showing. When we say what are the gametes, we are really saying which alleles does the parent have they could possibly pass on in their gametes. Now we are going to start by talking about what is called a monohybrid cross. Monohybrid. Mono means one. So we are only going to talk about one trait for now. We'll start talking about two traits at a time in our classes next week. But when we're doing a monohybrid cross, you should have a square that is divided into four. And then in each of the boxes, you will match up the letters or the alleles that come from each side of it. So here you can see there was a big G above, a little g to the side. I matched them up. And I would do the same thing for each square in my Punnett square. So you're always going to look which allele is listed to the left, which is above, and write them together in a box. So if I finish this Punnett square off, this is what I'm getting. Now in this particular sample, my male had two copies of the same allele. my female also had 2 copies of the same allele, I would call my male homozygous recessive. I would call my female homozygous dominant. That would be the types of alleles that they had. So when you use a Punnett square, you take a look at which alleles the 2 parents have, and you put them on each side and then match them up. Very frequently, we'll use a monohybrid cross as a control cross. A control cross is used to determine if a heterozygote is present or dominant homozygous. So frequently we will use a monohybrid cross, a cross on one characteristic, to try to figure out if an individual has 2 copies of the dominant gene or 1. Because to look at them, you wouldn't know. For example, as a general rule, having brown eyes is the dominant characteristic for eye color. But if I just look at you and see that you have brown eyes, I don't know if you have 2 copies of the dominant gene or 1 dominant and 1 recessive. There's nothing that shows me that. So a control cross is used to determine what does the organism have. So we'll do 2 examples. Let's say that I had a heterozygote. That would mean they had 1 of each allele. That'll be the first example. In the second example, we'll say it was homozygous dominant. In control crosses, we always use an organism that is homozygous recessive as the second parent. Since they only have the recessive gene, that's all they can pass on. So we'll fill in these 2 Punnett squares and determine what kind of ratio we get at the end. In Punnett square 1, I would fill in capital G, little g on the top, capital G, little g. So I'm looking what's to the left and what's above. In my bottom squares, I would put little g, and then little g, because that's what's to the left and above. So I would see 2 out of 4, which is the same as 50%, have the dominant phenotype. And I would see 2 out of 4, which is 50%, have the recessive phenotype. So half of them should show the dominant characteristic and half of them should show the recessive one. So if I had an individual and I didn't know if they were homozygous or heterozygous, I crossed them with a homozygous recessive parent and some of the offspring showed the recessive phenotype, then I could be sure, yep, parent 1 actually had the recessive gene. If, as in case 2, I have an individual that is homozygous dominant, here's what my Punnett square will look like. All 4 offspring possibilities would all show the dominant phenotype. So I would say 4 out of 4, or 100%, have the dominant phenotype. So I would know in this case that my parent didn't have the recessive allele. They had 2 copies of the dominant allele. So this is a way that you can use a Punnett square to figure things out. So I'm just going to pause.